Proteins, if you change their shape, you change their function. Changing the shape of a protein can easily be done through heating or changing of the pH. So a lot of these are proteins that are very specific and they are acting as enzymes, and enzymes have to click in to what they're trying to speed up, like a Lego fit. And if you start changing its shape, you're going to change its function. An example of this is sickle cell disease. Sickle cell disease is a blood disorder that is inherited, and the shape of the blood cell is supposed to be very spherical, sort of like the shape of a football. But with this one of the single amino acids gets substituted in the hemoglobin molecule. So maybe a valine gets substituted for an alanine. And this is something that happens genetically. And because we have changed the shape, it has changed its function, and this can be a lethal disease. All right, in addition to its primary structure, the physical and chemical conditions can affect the structure. So if you change the pH, if we make it more basic by adding salt, increasing the temperature or lowering the temperature, causes this protein to start unraveling. The term for this is denature. If you denature a protein, you're going to change its shape. If you change its shape, you're going to change its function. And it can become biologically inactive. So denaturation is something that happens first at the quaternary stage because your polypeptides are going to separate. And then if it keeps heating, the R groups are going to break their bonds, and then it's going to be down to the secondary configuration where you have your hydrogen bonds, and it's going to come out of its helix or it'll come out of its pleat, and then ultimately at the primary stage the peptide bonds are going to break. And this is what happens when you denature a protein. So you have completely rendered it inactive. This is one of the reasons why you can't run a temperature of 105 for very long, because at 105, we've heated up your body, and these enzymes are supposed to work within a certain temperature range, and you heat them up, and they're going to start unraveling at that quaternary shape, and before you know it, all of your enzymes have shut down, all of your hormones have shut down, your neurotransmitters have shut down, and of course, ultimately, that can lead to death. It's hard to predict the structure of a protein just from its primary structure. And so, there's a lot of stages that it goes through in order for it to be stable. There is a type of protein called a chaperonin, and chaperonins do just what it sounds like. They help. They assist in folding the protein into its right configuration. And sometimes when you have a misfolded protein, then you can have a disease such as Alzheimer's or Parkinson's. Mad cow disease is actually a prion. A prion is a misfolded protein where instead of folding it properly, the chaperonin is not doing its job, or it has been denatured, and the protein gets folded in the wrong configuration and can be an awful, awful disease, and especially with mad cow. Mad cow in humans, it's called Crutchfield-Jacobs. In sheep, it's called Scrapies, but it's a misfolded protein that gets in their feed, and they're all neurological disorders. I left this picture in your PowerPoint so that you can see that a fully assembled chaperonin, this protein, this is all wound together, has a little cap, and then the cylinder is hollow. And your polypeptide actually enters now this is the single polypeptide chain enters the cap. It enters the cylinder from one end, and then the cap attaches to it. And what happens is when the cap ultimately comes off, the protein is folded. So as long as this happens perfectly, we're good. And guys, if you really think about it, how perfect you really are. All of your proteins in your body are having to go through this process. And it goes from your DNA having the code out to the ribosome for this amino acid to be connected into a polypeptide. It'll end up going to, with the help of a chaperone, being folded properly. every single one of them has to be folded properly for you to be sitting there normal. And if it's not, then it usually result in a very serious neurological disease. Using X-ray crystallography is the best way to determine a protein structure. Another way is through an NMR, which is a nuclear magnetic resonance spectroscopy. This does not require for the protein to be crystallized. And bioinformatics is an approach able to predict the protein structure of sequences. But it's very hard, guys, because proteins are very, very complicated, and they can be just, a few amino acids long to these very, very complicated polypeptide chains.